And this presentation is going to be on FDA's regulatory program for chemical contaminants in foods. Some of you have been asking questions about this yesterday and today. And so here is your presentation that will address some of the questions that you've been asking. It'll be given by Dr. Paul South.
Thank you. Good afternoon. My name is Paul South. I'm a chemist at FDA's Center for Food Safety and Applied Nutrition in the Office of Food Safety. My responsibilities at FDA include developing policy, regulations, regulatory guidance, and compliance strategies on issues related to chemical contaminants in food. The title of my talk today is FDA's Regulatory Program for Chemical Contaminants in Foods. Today I will briefly discuss the statutory authority and enforcement options used by FDA to address chemical contaminants in food. I will also provide a description of FDA's monitoring program that addresses chemical contaminants, including FDA's Total Diet Study, as well as relevant FDA compliance programs. In addition, I will briefly discuss several of the chemical contaminants that FDA monitors in food, and finally, describe some of FDA's international efforts that address this issue. Federal Food, Drug, and Cosmetic Act provides FDA with regulatory authority with regard to adulterated foods that are in interstate commerce or delivered for introduction into interstate commerce. Specifically, Section 402 of the Act defines when a food is adulterated. Under Section 402A1, a food would be considered adulterated if the food bears or contains any poisonous or deleterious substance, and if the substance is added, FDA must show that the quantity of the substance in the food may render the food injurious to health, or if the substance is not added, FDA must show that the quantity of the substance in the food ordinarily renders it injurious to health. Therefore, if a substance is deemed added, then FDA need only show that it may render the food injurious to health in order to regulate consumption of the food containing the substance. The may render standard means that there is a reasonable possibility of injury to the consumer. However, if a substance is considered not added, FDA must go further and show that the substance would ordinarily render the food injurious to health before it can regulate its consumption. Under Section 402A1, FDA has different enforcement options to address chemical contaminants in food that affect public health. FDA's approach to addressing chemical contaminants with no FDA established levels is to handle instances of contamination on a case-by-case basis. For example, for chemical contaminants found in food, FDA conducts a health hazard evaluation by reviewing relevant information, including levels of the contaminant in the food, the type of food, consumption of the food, and the toxicological information for the contaminant to determine the level of a contaminant that would represent a potential health risk such that the food would be deemed adulterated and subject to enforcement actions under the Federal Food, Drug, and Cosmetic Act. Other options used by FDA to address chemical contaminants in food includes the use of guidance established by FDA, including guidance for FDA staff. This guidance includes action levels that are included in FDA compliance policy guides. Other guidance established by FDA is guidance for industry that includes guidance levels. Another enforcement option available to FDA but rarely used is the establishment of tolerances. This option has been used only once for addressing PCBs in food. One reason this option is rarely used by FDA is because it involves rulemaking, which is a long and often time-consuming process for both establishing, revising, and revoking tolerances. FDA's monitoring of food for chemical contaminants includes several different programs. One program is FDA's Total Diet Study. Other programs are FDA's Toxic Elements and Radionuclide Compliance Program, FDA's Pesticides and Industrial Chemicals Compliance Program, and FDA's Mycotoxin Compliance Program. The Total Diet Study, or TDS, sometimes called the Market Basket Study, is an ongoing FDA program that determines levels of various contaminants and nutrients in foods. From this information, dietary intakes of those analytes by the U.S. population can be estimated. Since its inception in 1961 as a program to monitor for radioactive contamination of foods due to fallout from nuclear weapons testing, the TDS has grown to encompass additional analytes. A unique aspect of the TDS is that foods are prepared as they would be consumed, referred to as table-ready, prior to analysis, so the analytical results provides the basis for realistic estimates of the dietary intakes of these analytes. The study designed for FDA's TDS involves the collection, preparation, and analysis of 280 foods and beverages. Using levels found in TDS table-ready foods and consumption amounts obtained from national dietary surveys, FDA calculates dietary intakes for chemical contaminants and nutrients for the total U.S. population, as well as for 14 age and gender population groups. Each sample is tested for approximately 400 analytes. Analytes measured under TDS include over 300 pesticides, 43 industrial chemicals, 13 radionuclides, and 18 elements, 4 toxic and 4 nutrient. Chemical contaminants that have been recently added to the program include dioxin in 1999, acrylamide, as well as perchlorate. For TDS, there are 4 regional market baskets collected each year. Each market basket is made up of 280 foods collected in 3 cities in each region. The 3 samples that are collected in each region are composited for each individual TDS food, then analyzed. TDS intake estimates are used for different purposes. For example, they are used to provide estimates of background dietary exposure, to monitor the impact of regulatory actions, to identify potential health hazards, to assess national nutrition monitoring efforts, and to provide support for international development of food standards and risk assessments. In the past, FDA's TDS has identified elevated levels of certain chemical contaminants in foods. For example, in 1996, an apple prune juice sample was found by FDA to contain elevated levels of lead. After tracing the samples back to the collection site, FDA determined that the source of lead in the juice was a prune juice concentrate used in the manufacture of the product. In another incident in 1996, elevated levels of lead were found in samples of chicken and vegetable baby food. Again, after tracing samples back to the collection site, FDA determined that carrots used in the product had been grown in a field that had been previously treated with a pesticide containing lead arsenate. Other examples involving unusual levels of arsenic were in 1997, when elevated levels of arsenic were found in baby food carrots, and in 1998, elevated levels of arsenic were found in peanut butter. One chemical contaminant that FDA monitors is acrylamide. Acrylamide is a chemical contaminant that is formed in certain foods during processing. Specific processes associated with acrylamide formation include frying, baking, or broiling. Acrylamide can be found in a variety of foods, including potato chips, French fries, bread, cereal, sweet potatoes, asparagus, cocoa, and coffee. Levels of acrylamide in food can vary in production lots, between different products, and between different food types. FDA has measured levels of acrylamide in different foods and has posted data on acrylamide levels in approximately 2,600 food samples. FDA has posted estimates for acrylamide intake. The following table shows some of the top 20 foods by mean acrylamide intake. The intake levels are a function of both the acrylamide level found in the food as well as the amount of the food consumed. This table is available on our website. FDA has also been involved in research to study the process by which acrylamide is formed in food. For example, FDA has studied the effect of cooking temperature on acrylamide formation in French fries. FDA studies indicate that in French fries, acrylamide formation increases with higher frying time and temperature. In addition, acrylamide formation is highly correlated with darker French fry color. Similarly, in toasted bread, light toasting causes very low acrylamide formation, while dark toasting causes low to moderate acrylamide formation. In addition, scraping toast to remove brown surfaces has been found to reduce acrylamide levels. Another chemical contaminant FDA monitors in food is arsenic. Inorganic forms of arsenic, including the tri- and pentavalent forms, are the arsenic species of greatest toxicological concern. In contrast, organic forms of arsenic, which is the predominant form present in fish, demonstrate little toxicological potential. Recent concerns have been raised regarding the occurrence of inorganic arsenic in certain foods, including apple juice, pear juice, and rice. Another chemical contaminant that FDA monitors in food is dioxin. Dioxin and chemically related compounds, referred to collectively as dioxin-like compounds or DLCs, can be found in food-producing animals. Because DLCs tend to accumulate in fat, consumption of animal-derived foods, including meat, poultry, eggs, fish, and dairy products, account for approximately 95% of human exposure. Studies suggest that DLC exposure may lead to a variety of adverse health effects, including reproductive and developmental problems, cardiovascular disease, increased diabetes, and increased cancer. Reduction of emissions from numerous combustion processes have resulted in a significant decline in levels in the United States. EPA estimates emission reductions to be more than 90% since 1997. Since 1995, FDA has been monitoring dioxin in different foods. In 1999, FDA began annual monitoring for dioxins as part of FDA's Total Diet study. In addition to TDS samples, FDA analyzes non-TDS foods or targeted foods and animal feed in an effort to gather additional information on sources and pathways for dioxin in the diet. FDA has posted data for dioxin levels in TDS and non-TDS foods and posted exposure estimates using these results. The following graph provides exposure estimates for dioxin in TDS foods for the total U.S. population and for 14 age and gender population groups. The Environmental Protection Agency recently released a dioxin hazard assessment addressing non-cancer health effects associated with dioxin exposures. Included in the EPA dioxin hazard assessment is a reference dose of 0.7 picograms per kilogram body weight per day. FDA has posted a dioxin questions and answers document on the FDA website that provides general information on dioxin and the food supply. Another chemical contaminant that is monitored by FDA is lead. There is little to no margin of safety for lead and therefore some level of risk associated with even low levels of exposure. Lead is of particular concern for the developing fetus because the placenta presents a minimal barrier to the movement of lead from maternal to fetal circulation. In addition, in infants and younger children, food consumption per body mass is greater as compared to adults. Another concern with lead is that children absorb lead more readily than adults by a factor of about 4. Moreover, the immature status of developing organs, including the brain, kidneys, and liver, make the fetus, infants, and children more sensitive to the toxic effects of lead. FDA's TDS has been analyzing food for lead for decades, and TDS results, as shown in this slide, indicate a large reduction in dietary lead intake. A number of factors, including FDA's ban on the use of lead-soldered cans for food, are thought responsible for this decrease in dietary lead intake. In 1994, the California Department of Public Health identified imported Mexican candy contaminated with lead that had migrated from ink used on the candy packaging. In addition to the package, FDA also discovered that Mexican candy made with certain ingredients, for example, chili and tamarind pulp, contained a higher lead level than that found in domestic candy. To address this issue, in 1995, FDA issued its first guidance level for lead in candy of 0.5 ppm. In 2006, FDA issued a revised guidance level of 0.1 ppm for lead in candy likely to be consumed frequently by small children. Another chemical contaminant that FDA monitors in food is perchlorate. Perchlorate is both naturally occurring and man-made. For example, it is found naturally in certain fertilizers, but it is also manufactured for use as a propellant in rockets and missiles. Perchlorate has been detected in numerous surface and groundwaters in the U.S., including the Colorado River. One issue associated with perchlorate is that it can inhibit iodide uptake by the thyroid. Impairment of the thyroid can affect the fetus and newborn, resulting in delayed development and decreased learning capacity. In 2005, the National Academy of Sciences and EPA established a reference dose of 0.7 micrograms per kilogram body weight per day, based in part on its role in the inhibition of iodide uptake. FDA has been testing TDS and non-TDS foods for perchlorate since 2006. The following table provides exposure estimates generated from TDS foods for different age and gender population groups. FDA has also monitored food and feed for melamine. In 2007, FDA developed a safety risk assessment in collaboration with USDA's FSIS in response to the deliberate contamination of vegetable protein products imported from China that were mislabeled as wheat gluten and rice protein concentrate. The assessment described the risk to human health associated with eating pork, chicken, fish, and eggs from animals fed animal feed adulterated with melamine and its analogues. Results of the safety risk assessment indicated that consumption of pork, chicken, domestic fish, and eggs from animals consuming feed contaminated with melamine and its analogues was very unlikely to pose a human health risk. FDA also analyzes food for mycotoxins. Mycotoxins are low molecular weight secondary metabolites produced by certain fungi during the growing, processing, transport, and storage of certain foods. Mycotoxins occur naturally and are difficult to avoid. Various toxicological outcomes occur from consuming mycotoxins. Many mycotoxins are stable to heat and food processing procedures. FDA monitoring activities consist of targeted sampling of foods that are susceptible to mycotoxin contamination. Through this monitoring program, FDA has derived estimates on the incidence, levels, and dietary exposure that are used for risk assessments and for establishing guidelines. To address mycotoxins in food, FDA has established limits, including action levels and guidance levels for different mycotoxins in food. For example, for all products except milk, FDA has established an action level of 20 part per billion for a mycotoxin called Aflatoxin. For milk, FDA has established an action level of 0.5 part per billion for a specific aflatoxin called Aflatoxin M1. These action levels are found in FDA compliance policy guides in section 527-400, 555-400, and 683-100. FDA has also established an action level of 50 parts per billion for a mycotoxin called patulin in apple juice and apple juice products. This action level is found in FDA Compliance Policy Guide, section 510-150. FDA has also established a guidance level for a mycotoxin called deoxynivalenol in finished wheat products of 1 part per million. This guidance level was established by FDA in a letter that was sent to state food officials and feed and grain trade organizations in 1993. Established a guidance level for a mycotoxin called fumonisin in different corn products that ranges from 2 to 4 parts per million. These levels are contained in an FDA guidance document posted on the FDA website as indicated. In addition to these monitoring efforts, FDA also participates in international committees to address chemical contaminants in food. For example, FDA participates in the Codex Alimentarius Committee on Contaminants in Foods, a committee established under the Codex Alimentarius. Codex was created in 1963 by FAO and WHO to develop food standards, guidelines, and related standards program. The main purposes of this program are protecting the health of consumers, ensuring fair practices in trade, and promoting coordination of all food standards work undertaken by international government and non-government organizations. The Codex Committee on Contaminants in Food establishes maximum levels for contaminants and naturally occurring toxicants in food and feed. For example, maximum levels established by this committee have addressed melamine in food and feed, as well as lead in fruit juice. The committee also elaborates codes of practice for prevention and reduction of contaminants and naturally occurring toxicants in food and feed. Recent codes of practice developed by the committee include a code of practice addressing lead contamination in food and a code of practice addressing dioxin and dioxin-like PCB contamination in both food and feed. Finally, the committee prepares priority lists of contaminants and naturally occurring toxicants for risk assessment by the joint FAO and WHO Expert Committee on Food Additives. While not officially part of the Codex structure, the joint FAO and WHO Expert Committee on Food Additives provides independent scientific expert advice to the Commission and its specialist committees like the Codex Committee on Contaminants in Food. With that, I'd like to conclude and thank you for your attention today. I note that more information on FDA's regulatory program for chemical contaminants in food is found on FDA's website, or if you have specific questions, you can contact me, paul.south@fda.hhs.gov. Again, thank you, and I'd be happy to answer any questions.
Thank you very much for the presentation. There are several questions. As we always, we take them in the order that they are listed, and I'll just read them and you can respond. Are herbicide levels tested for in products made from GMO Roundup Ready crops?
EPA actually has established tolerances, which is a little different than chemical contaminant tolerances. Tolerances for pesticides are established by the EPA, and then they actually are looked at by FDA. So for these certain herbicides, there would be pesticide tolerances established for crops.
Okay. The next question is, foreign manufacturers constantly ask what permissible levels of lead, et cetera, what they are. How can they know if they are within the limits?
That's a good question. We actually, and I mentioned in my presentation that we do have a guidance level for lead in candy consumed by children, but certainly lead is an issue for other products as well, and certainly for as I mentioned, children, kids that consume certain products in high levels. FDA would look at these levels of lead in these types of products on a case-by-case basis, but if companies would like to look for some other advice on what lead levels would be considered levels that are found internationally, they could go to the Codex Alimentarius. That's on the website, and there's actually a set of standards for general contaminants in food, and they could go there, and there are a listing of levels for lead in different food products. So, and that would be on the Codex website.
All right, I see we have two questions that are visible to me on acrylamide, and I think maybe I'll just ask them both and you can respond, because they're both the same. What does acrylamide do to you? How much is too much? I love burnt toast. What about levels of acrylamide in burnt popcorn?
Well, one of the issues with acrylamide is that it is a carcinogen, so it can cause cancer. In regard to anything burnt, we have looked at the effects of browning on certain types of food, and we have found that increased browning or increased color formation does result in higher levels of acrylamide. It's a good question in regard to what happens to those levels. I imagine, popcorn, before it gets burned, it probably turns brown, and then you probably have that acrylamide formation. So certainly I would suggest if you're worried about acrylamide, don't burn your popcorn.
All right, the other question is, on average, how many foods are tested per year?
We had mentioned, so we do have different programs. The total diet study, for those products, there's 4 market baskets, and each market basket, as I mentioned, is in different regions of the country, and there's 3 cities that are chosen per region, and those actually vary per year, so it's not going to be the same cities in that region. So it'd be 280 foods, and actually the 280 foods are composites of 3 samples, so that's from 3 cities, but they would be composited, and then we would analyze that one composited food sample, and in 4 different regions, so it would be 280 times 4. That's for the total diet study. But again, we have other compliance programs that analyze food as well, and in regard to those different, it depends on the food and the compliance program. But in regard to how many foods, we're talking thousands of different foods. And then in addition, there's other compliance programs that go in and do inspections, and foods are gathered there. So it's difficult to really provide you with a good number on how many foods, but we're talking thousands of foods.
All right, this is our last question because we're going to move on to our next presentations. But the one is, are manufacturers required to test for any of these chemicals?
At this point, they aren't required to test for these different chemicals. It would be up to the company to test themselves, but if FDA did some testing and found those levels. But again, it's not a requirement for companies to test for these contaminants. Now, of course, the Food Safety and Modernization Act is, obviously that has passed, and there is some requirements to address chemical hazards as well as microbiological and physical and radiological. So that will have some impact on the requirements for manufacturers.
All right, well, thank you very much. We're going to prepare for our next presentation, which will start in about 60 seconds.